Hello and welcome. In this webinar, I will be introducing the new Immunotherapy module that extends our existing virtual tumour technology. I will start with a short introduction to Physiomics and will then move on to give a brief overview of our virtual tumour platform, which is a unique predictive tool for optimizing dosing and scheduling of anti-cancer combination regimens. There is a great deal of interest at present in combining immunotherapy agents with conventional anti-cancer therapies, and I'll move on to discuss the problems inherent in optimizing these combinations. Finally, I'll present our solution to these problems in the form of an extension to our virtual tumour technology that allows optimization of novel immunochemotherapy regimens. Physiomics was founded in 2001 and listed on the AIM market in 2004. We are based in Oxford, in the UK. Our focus is the development of predictive models with the aim of improving the success rate of drug discovery and development, reducing timelines, saving money, and, ultimately, making better decisions. We have a particular focus in oncology with our unique virtual tumour technology. However, we do offer a range of products and services across the drug discovery and development pipeline. In addition to the virtual tumour, these include DrugCard, our combinations and regimens database aimed at both researchers and clinicians, and EasyApp, our cardiac toxicity prediction service, in addition to standard PK/PD modelling. To date, we have successfully completed over 50 commercial projects and have 3 large pharma customers, in addition to several biotech and specialty pharma customers. We have received grant funding from a number of sources, including a grant from the Technology Strategy Board to support the initial development of the clinical version of the virtual tumour. This significant extension to our virtual tumour capability was developed in collaboration with 2 academic groups, and we are now working with Merck Serono on a long-term VT clinical project. Our latest advance has been the development of an immunotherapy module for the virtual tumour, which will be the focus of today's webinar. Before we move on to look at the specifics of adapting our VT platform, modelling, predicting and optimizing the efficacies of anti-cancer immunotherapies, I will briefly describe the fundamentals of the VT technology. The virtual tumour was developed in response to the obvious drawbacks of the standard drug discovery approach, which relies heavily upon preclinical experimentation and hence is costly and time-consuming and provides little opportunity for optimization of dosing regimens. In contrast, our approach requires only limited preclinical data to provide optimized dosing schedules at reduced cost and in a much shorter time frame. So, how does the virtual tumour work? The virtual tumour is an agent-based model of a heterogeneous population of growing tumour cells. Each cell or agent within the population runs an individual instance of the model. The model represents key tumour properties, including growth dynamics and spatial characteristics, but does not try to replicate the full complexity of known tumour biology. We are concerned with the overall effect of a therapy on the fate of the tumour cells, be that to cause cell cycle arrest or cell death, or some combination of these effects. Taking this high-level approach, we have been able to build the efficacy of an individual tumour of a wide range of anti-cancer therapies, including traditional chemotherapeutics, targeted agents, biologics, and irradiation. The model is calibrated using 3 types of data, these being control, that is untreated, tumour growth curves for the cell line of interest, PK data, either plasma or tumour, for the compound or compounds of interest, and tumour growth inhibition data for the individual compound or compounds as single agents. If available, additional biomarker data can also be used, but these are not essential. From this limited set of data, we can predict optimal dosing schedules for both monotherapies and combinations. Unless some mechanistic synergy between 2 therapies is expected, we do not normally require tumour growth inhibition data for the combinations in order to predict their efficacies. Our standard model output is shown on the right of this slide. Experimental data for individual animals are represented by the grey lines, while the experimental mean and standard error are plotted in black. Our model prediction is overlaid in red, and the confidence interval for the prediction is given by the pink shaded area. The preclinical version of the model predicts the change in mean tumour volume over time, and over 40 studies have confirmed the predictive power of this. Clinical model predicts the change in mean tumour diameter over time for all lesions. In carrying out a clinical translation, our approach is unique in that we take into account both clinical PK and the difference in tumour growth dynamics between a preclinical model and a clinical indication. In this way, we have successfully translated the efficacies of combinations of small molecules such as selumetinib, vemurafenib, and docetaxel. If you would like more detailed information, then presentations and posters giving details of both preclinical and clinical VT case studies are available on our website. I'm now going to move on to the main focus of today's webinar, how the virtual tumour can be applied to predicting and optimizing immunotherapy efficacy, particularly in combination with conventional anti-cancer therapies. Increasing evidence has shown that a number of conventional anti-cancer therapies, including radiotherapy, cytotoxic, and targeted agents, promote specific anti-tumour immune responses that contribute significantly to their therapeutic effects. In addition, analysis of clinical tumour biopsies suggests that tumours with pre-existing active immune microenvironments respond better to immunotherapy. Hence, combining conventional therapies with immunotherapies should provide improved anti-tumour efficacy. However, dosing and scheduling of conventional therapies combined with immunotherapy need to be optimized to take full advantage of a limited window for immunopotentiation while avoiding immunosuppression. Many conventional anti-cancer therapies have been shown to induce an immune response, and just some are listed here, including irradiation, platins, anthracyclines, taxanes, and targeted agents. There are 4 overall mechanisms by which chemo and radiotherapy can activate a tumour-targeting immune response. By directly modulating the functions of innate or adaptive immune cells. 2. Subverting the immunosuppressive mechanisms established by cancer cells. 3. Enhancing the immunogenicity of cancer cells or their susceptibility for immune effector mechanisms. And 4. By inducing immunogenic cell death. This is a form of cell death in which dying cancer cells function as a vaccine that promotes a tumour-specific immune response. Some agents induce multiple effects through multiple mechanisms. For example, gemcitabine, which selectively depletes myeloid-derived suppressor cells, preferentially inhibits B-cell proliferation, T cells, and promotes the activation of function of effector T cells, restores tumour antigen cross-presentation by dendritic cells, and increases class I HLA expression. Targeted agents can also provoke an immune response through a variety of mechanisms. On the other hand, irradiation provokes an immune response largely through inducing immunogenic cell death. It has been suggested that all these mechanisms may rely on the capacity of dendritic cells to engulf, process, and present tumour antigens to effector T cells. However, this capacity is impaired in tumour-bearing animals and cancer patients, which is one of the most significant factors in the failure of immunotherapy. Adjuvant radio or chemotherapy to boost the immune response might therefore play an important role in ensuring the success of immunotherapy. But, since the immunological effects of conventional therapies are dose and time dependent, rational design and optimization of the combination therapy schedule is crucial. Among the requirements for successful immunochemotherapy, there is a general consensus that appropriate dosage, timing, and sequence of administration are likely to be critical. But what does an optimal schedule for immunochemotherapy look like? In fact, the optimal regimens for immunotherapy and radio or chemotherapy combinations remain to be established, and there are several considerations to take into account. Firstly, as different cytotoxic agents have different immunological effects, optimal treatment schedules may vary. Moreover, the majority of conventional therapies at standard clinical doses actually have immunosuppressive effects. And, although both dosage and order of administration have been shown to influence the success of immunochemotherapy, there are contrasting reports in the literature. Some studies suggest that chemotherapy should be given first, while others suggest that the immunotherapy should be given first. Different tumor types and/or immunochemotherapy regimens therefore behave differently, and appropriate therapeutic regimens must be designed in each case. We have already validated our virtual tumor platform through numerous commercial and internal studies as a tool for optimizing conventional anti-cancer combination regimens. Adapting the technology to also allow optimization of immunochemotherapy regimens seemed an obvious progression. We therefore extended our virtual tumor model with the development of an immunotherapy module, and validated this extended model by carrying out an internal case study based on data taken from the literature. We decided to investigate the capability of the model to accurately predict the efficacy of an immune checkpoint blocker, specifically an anti-PD-L1 antibody, combined with irradiation. Our starting point was published PK and tumor growth inhibition data for the anti-PD-L1 antibody and irradiation, with which we calibrated separately the efficacies of the two monotherapies. We also carried out an extensive survey of the literature to gather data for calibrating the immune response to irradiation and the likely combination synergy that would arise with the antibody. The outcome of the study was that we were able to correctly predict the efficacy of the immune checkpoint blocker and irradiation combination in two syngeneic mouse models. The basis of the model is our standard VT model, in which a population of tumor cells is acted upon directly by a cytostatic or cytotoxic agent, in this case irradiation, resulting in cell cycle arrest or cell death. Immunotherapeutics do not act directly upon tumor cells, but through a variety of mechanisms, they promote an anti-tumor immune response. In our case study, immune checkpoint block by the anti-PD-L1 antibody increases activation of effector T cells, which then attack the tumor cells. These mechanisms account for tumor growth inhibition by the individual agents, but cannot account for any synergy between the agents. This synergy arises through immunogenic cell death induced by the irradiation. This form of cell death promotes maturation of dendritic cells and thereby stimulates the anti-tumor response of the activated T cells. The synergy decays over time as tolerance signals are released by the dead tumor cells. The structure of this synergy model, the dynamics of the immune response, and the magnitude of the resulting immunopotentiation are derived from published data. As this immunotherapy is an extension to our standard virtual tumor model, the data requirements for calibrating the efficacy of a monotherapy are essentially the same. In an ideal situation, giving us the highest level of confidence in our predictions, we would use tumor PK profiles with cell cycle and immune response biomarkers, in addition to the obligatory control and treated tumor growth curves. However, for our case study, we only had limited data taken from the literature, consisting of plasma PK profiles for the antibody, along with tumor growth curves in two syngeneic mouse models. In a typical VT project, synergy between cytostatic or cytotoxic agents arises through their direct effects on tumor cell fate, and this will automatically be reflected by the model. However, in this case study, synergy is arising through quite a different mechanism, involving indirect effects on immune cells within the tumor microenvironment. We had no immune response data with which to calibrate the magnitude of this indirect synergy. We once again looked to the literature. We carried out a comprehensive survey of combinations of similar immunomodulators with agents that induce immunogenic cell death, and discovered that such combinations are typically up to 3 times as effective as the monotherapies, and commonly around twice as effective. We therefore assumed a level of synergy consistent with these findings for our anti-PD-L1 irradiation combination, calibrated the model accordingly, and made our predictions. Here you can see our model calibrations for the control, antibody, and irradiation tumour growth curves in the two syngeneic mouse models. In this case, the red lines represent a simulation, as we are not yet making any predictions, just tuning the VT model to the untreated tumour growth dynamics and the efficacies of the monotherapy. Finally, we can show you the outcome of our study. As you can see, the virtual tumour immunotherapy model has made an accurate prediction of the combination efficacy in both mouse models. In summary, the case study demonstrates that we have successfully extended our virtual tumour capability to making efficacy predictions for an immune checkpoint blocker combined with an agent that induces immunogenic cell death. This model has also been validated in a separate study with a commercial partner. The model is a high-level abstraction of the biology, but tracks T-cell activation, dendritic cell maturation, and immunopotentiation. The focus of the model at present is adaptive immunity and immunogenic cell death. It can readily be adapted or extended to simulate other immunotherapy modalities and combinations. Thank you for listening, and we would welcome your questions. Please contact us for further information.